So this is the second session of development of brain, spinal cord and pituitary gland. So here we'll see the development of spinal cord and neural tube defects. So the parts of the neural tube caudal to the hindbrain forms the spinal cord. It is in 4 phases, that is formation of mantle marginal layer, formation of basal and alar plates, histogenesis of the cells and positional changes of the spinal cord. You can see here this is the neural tube lined by the neuroepithelial cells. Neuroepithelial cells will differentiate to form the ependymal cells lining the central canal, and then the mantle layer, and then the marginal layer. So this mantle layer will further differentiate into alar plate dorsally and basal plate ventrally. The basal plate forms the ventral horn and the alar plate forms the dorsal horn. So stages in the formation of the spinal cord, you can see the formation of mantle and marginal layers, proliferation of neuroepithelial cells. They differentiate to form the ependymal, mantle and marginal layer. So this will be the mantle layer, this will be the outer marginal layer, and this will be the ependymal layer. So basal and alar lamina. So sulcus limitans will divide the mantle layer into the dorsal alar plate and ventral basal plate. And this basal plate will form the motor neurons, whereas the motor horn, and the dorsal plate forms the sensory horn of the spinal cord. So histogenesis of cells. So neuroepithelial cells will form neuroblasts, which form neurons, spongioblasts, which form astrocytes and oligodendrocytes, and the mesenchyme will give rise to microglial cells. So neuroblasts, initially they form apolar neuroblasts, followed by bipolar neuroblasts, followed by multipolar neuroblasts. Then positional changes in the spinal cord, you can see here. In 3rd month, the spinal cord extends throughout the vertebral canal. In the 5th month, it extends up to S1. And in the newborn, you can see it is at the level of L3, and in adults, it is at the level of L1. So what changes will happen when the spinal cord recedes? So spinal nerves do not arise at the level of corresponding intervertebral foramina. Spinal nerves run obliquely downwards to reach corresponding intervertebral foramina. Lower portion of pyometer forms fibrous band called phylum terminal that extends from lower end of spinal cord up to the first coccygeal vertebrae. Dura mater extends up to second sacral vertebrae. The pyometer is derived from the neural crystals, whereas arachnoid matter and dura mater are derived from the condensation of piscine that surrounds the neural tube. So functional columns of the spinal cord. In the spinal cord, there are only 4 functional columns. So in the basal lamina, there are 2 functional columns. General somatic efferent, that means provides innervation to skeletal muscles. General visceral efferent, that means provides innervation, motor innervation to the viscera. Then in the alar lamina, there are 2 columns. General visceral afferent, so sensations from the viscera, and general somatic afferent, exteroceptive and proprioceptive sensations. Now, myelination. So the process of formation of myelin around the axons. In the CNS, it is by oligodendrocytes. In the PNS, it is by Schwann cells. So begins during 4th month of intrauterine life and continues after birth up to 2 to 3 years. Motor fibers myelinate earlier than sensory fibers. Myelination of corticospinal tract begins at 9th month of intrauterine life and continues up to 2 years after birth. So you can see here, this is the axon, this is the Schwann cell, which is myelinating, or which is forming the myelin sheath. So neural tube defects. So these are the group of conditions formed owing to the non-closure of neural tube. Closure of neural tube, as I said earlier, cranial neuropore closes by 25th day and caudal neuropore by 27th day. So these are the commonest birth defects. Occurs due to deficiency of folic acid, which is important for DNA and RNA synthesis, cell division and myelination. Folic acid supplementation to the mother reduces the development of neural tube defects. The main defect is in the methylene tetrahydrofolate reductase enzyme deficiency, so which is associated with the neural tube defects. So these are the commonest neural tube defects. Spina bifida, which is of 3 types: occulta, meningocele, meningomyelocele. Then anencephaly, encephalocele, anencephaly and rhachisis. You can see here, spina bifida is categorized into 3. So first it is spina bifida occulta, where bifid spine is not visible on the surface, the side covered by hairy skin. Then spina bifida cystica, which is having 2 parts, meningocele and meningomyelocele. Meningocele means only meninges will come out, whereas meningomyelocele, both meninges and spinal cord will come out. So prevention, folic acid supplementation, and it can be diagnosed by maternal serum fetoprotein. So you can see here, this is a normal one. So you can see this is the spina bifida occulta, so it is hidden. So with the meningeal sac, meningocele, with meninges and spinal cord, it is meningomyelocele. So you can see here, this is the spina bifida occulta. And what is diastematomyelia? So that means there is a bony plate. You can see here, this is the bony plate which is splitting the spinal cord into 2. So this is also called a split cord malformation. You can see here, spina bifida occulta, meningocele and meningomyelocele. Anencephaly, encephalocele, anencephaly and rhachisis. So in rhachisis, the neural plate will not form the neural tube. Neural plate will remain like neural plate. Okay. So you can see here, anencephaly is the failure of closure of anterior neuropore. So there is absence of major portion of the brain. So encephalocele, failure of formation of the skull cap, so through which there is protrusion of brain and meninges. Anencephaly, defective occipital bone, spina bifida of the cervical vertebra, retroflexion of the neck. Rachisis, failure of closure of the posterior neuropore, or even the neural plate itself will remain like plate or groove, so that there will be flat nervous tissue exposed to the surface. You can see here. So this is the rachisis. If this also involves the brain, so then we call it as craniorachisis. Hydrocephalus, it's the excess of CSF in the ventricular system. Causes may be obstruction of CSF passage or excess production of CSF. Impaired communication between ventricles and subarachnoid space. So dilatation of ventricular system results in separation of the cranial bones. It produces large head size and degeneration of nervous tissue. Enlargement of central canal spinal cord is also called as syringomyelia. So I have already dealt with it in the first class of the nervous system. So now you can see here, this is the development of spinal nerve. The spinal nerve will have the motor fibers and sensory fibers. If you see the sensory fibers are derived from the dorsal root ganglion neurons, which are derived from neural crest, whereas motor fibers are derived from the anterior horn cells, which develop from the basal lamina. And you can also see this white and gray rama communicantes and the sympathetic ganglion, which are derived from the neural crest cells. So the sensory fibers are derived from the neural crest cells, whereas the motor fibers of the spinal nerve are derived from the anterior horn cells, which develop from the basal lamina. So functional columns from the brainstem. So in the spinal cord, we have seen only 4 functional columns, namely the general somatic efferent and general visceral efferent, and general somatic afferent and general visceral afferent. But here in the brainstem, there will be 7 functional columns, as you can see here. In the basal lamina, you can see 3 functional columns: general somatic efferent for striated muscles of head that are not originated from pharyngeal arches, special visceral efferent for the striated muscles which develop from pharyngeal arches, and also for trapezius and sternocleidomastoid, and general visceral efferent, which are preganglion parasympathetic fibers to the viscera in the head neck region. So in the alar lamina, so there are 4 columns, namely the general visceral afferent, general sensation from the viscera; special visceral afferent, taste sensation from the tongue; general somatic afferent like proprioception, tactile sensation from face and oral, nasal and pharyngeal mucosa; and special somatic afferent for auditory and vestibular impulses. Now you can see here, so these are the 7 columns. Okay, in the spinal cord there are only 4, but in the medulla or in the brainstem there are 7 columns. From medial to lateral, general somatic efferent, special visceral efferent. So general somatic efferent is just motor to the viscera. Special visceral efferent is the branchiomotor, that is to the pharyngeal arch muscles. General visceral efferent. General somatic efferent is just motor to the skeletal muscles, which are not developing from pharyngeal arches. General visceral efferent is motor to the viscera, whereas special visceral efferent is to the branchiomotor, that is the skeletal muscle which develop from the pharyngeal arches. So general visceral afferent is general sensations from the viscera. Special visceral afferent is taste sensation from the tongue. So general somatic afferent is extraceptive and proprioceptive sensations, and also touch, pain and temperature. Special somatic afferent is hearing, vestibular and cochlear. So let me show you. You can see here, this is the general somatic efferent to the skeletal muscles which are developed other than the branchial arches. Special somatic afferent is striated muscles of the pharyngeal arches. Then general visceral efferent to the visceral smooth muscles, it is in the viscera. And general visceral afferent is general sensations from the viscera, and special visceral afferent is special sensations like taste sensations from the tongue. Then general somatic afferent is from the skin, and special somatic afferent is from the vestibulocochlear nerve. So you can see here, general somatic afferent is oculomotor nucleus, trochlear and absent nucleus, which supply the extraocular muscles of the eyeball, which are not developed from the pharyngeal arches, and hypoglossal nucleus supplying the tongue, which is also not developed from the which have muscles which develop from the occipital myotomes. So special visceral efferent have the motor nucleus of trigeminal nerve, motor nucleus of facial nerve, and nucleus ambiguous, which supplies the muscles which develop from the pharyngeal arches. So general visceral efferent column: Edinger vestibule nucleus, lacrymated nucleus, superior salivatory nucleus, inferior salivatory nucleus, and dorsal nucleus vagus. All these supply the viscera. General visceral afferent and special visceral afferent, nucleus of tractus solitarius, both general as well as special sensations, that is taste sensation also comes here. General somatic afferent include the chief mesencephalic and spinal nucleus of the trigeminal nerve. So mainly the trigeminal nerve. So here, special somatic afferent the vestibular cochlear, vestibular nuclei and cochlear nuclei. So these are the 7 columns: general somatic efferent, special visceral efferent, general visceral efferent, general visceral afferent, special visceral afferent, general somatic afferent, and special somatic afferent.